Hello class, welcome back to NEET Dude JN classes, and in this today's lecture we are going to take the biotechnology, the second class. Actually I have given about the I have already posted about the biotechnology in my previous lecture, and today we are going to discuss about the further concept. Before going into the further concept, just we have to recall, and those who not listen that lecture, please go and watch that lecture. If you listen that concept, this concept will be very clear. First thing that you have to remember in this biotechnology is preparing the recombinant DNA. That is the main thing. After preparing this recombinant DNA, what we will do? For what purpose we are preparing the recombinant DNA? That is secondary part. Later we will discuss. The first thing is we have to prepare the recombinant DNA. This is the main thing. We have to prepare the recombinant DNA. This recombinant DNA simply we can represent as rDNA. rDNA. R stands for recombinant. DNA means deoxyribose nucleic acid. So this is the main thing we have to prepare. In this introduction, in this chapter, we are going to prepare this rDNA. After preparing this rDNA, what we will do? That we will discuss later. This recombinant DNA is also called with another name. This recombinant DNA, rDNA, okay, rDNA it is. Now we listen. To prepare this recombinant DNA, in my previous class, we discussed about the isolation of DNA. Yes. We have to isolate the DNA. First step is isolation of DNA. Isolation of DNA. Yes, you know, different cells we treated with a different cell. You can take the animal cell DNA, you can take the plant cell DNA, you can take the fungal cell DNA, or you can take the bacterial cells DNA. Any DNA you want, you take. You treat it with the respective enzymes, and finally all the products are broken down and we are taking the DNA outside. This process of taking of DNA outside is called what? Spooling. That is, I taught in my previous lecture. You go and watch that. Yes, to precipitate this DNA inside of the test tube, we have to add the chilled ethanol. Yes, after this isolation of DNA, next concept we discussed about the cutting the DNA. A long DNA we got now, we have to cut that long DNA. Why? Because we require only a small piece of the DNA, which we call it as a gene of interest. We don't want such long polymer, we want only the small gene of interest. For that purpose, by using the restriction enzymes or a molecular scissors, we cutted the DNA into number of what they are, fragments. Just I am recollecting those terms. Fragmentation of DNA. Yes, we make it into number of fragments. Now you got the number of fragments. And these fragmentation that you will get by the cutting the DNA with the restriction enzymes, exonuclease and endonuclease. In past class it is there. Now in this all fragments, we don't want all the fragments, we want only one fragment. We want only one fragment. For that purpose only, we treated with the porosis. What porosis? Gel electroporosis. You know what gel that we are used. The gel that is used is agarose gel. You know that agarose gel is a natural polymer which is extracted from the seaweed. From seaweed they have taken, from ocean it is taken. This natural polymer. In this electric field is there, cathode and anode, and we are pouring all the DNA fragments in a well and switch on the electric field. When we switch on the electric field, these DNAs are what charged molecule? Negatively charged molecule. These negatively charged molecules are moving towards anode. How they are moving? According to their size. Small the DNA fragment size, far it will go. Larger the DNA fragment size, there it will be present there only. It will be near to cathode. If it is small, means it will be near to anode. To see these DNA fragments which are distributed in the gel electroporosis, we have to add what? Chemical. We have to add the chemical ethidium bromide. After adding the ethidium bromide, we have to pass the radiation. What radiation? UV radiation. At that time, what color bands we can see? Bright orange colored bands we are seeing, which are distributed in the gel. In these bands, we have to take only one fragment, that fragment which we require. Yes, we can take that one fragment. This process of taking of that desired DNA fragment or gene of interest, it is called what? Elution. There is a difference between the spooling and elution. The taking the entire DNA is spooling, and taking the only gene of interest is elution. This are the three parts that we discussed, 3 points that we discussed in my previous lecture. Those, if you are not watched that, go and please watch how we taken the DNAs of different plant cells and all those. Now finally we got the gene of interest. Just remember, by the process of elution we got the gene of interest. Just remember, this is our gene of interest. This is our gene of interest. This is what we want. The main theme is I have to prepare the recombinant DNA, which is known as rDNA. For that purpose I have the gene of interest now. It may be animals DNA or plants or anywhere, it is gene of interest, that's all. Now this gene of interest, the name number 1 is, it is gene of interest. This gene of interest is also called foreign DNA. This gene of interest is also called foreign DNA. Foreign DNA. And this foreign DNA is also called with another name. The another name is gene of interest, or it is also called foreign DNA, or it is also called, I forgot, later I will tell. Now remember, gene of interest, which is also called foreign DNA. Yeah, what we are going to do, yeah, I remembered, that is called, sorry, this screen is making some trouble. Yeah, I remembered that. That foreign DNA is also called alien DNA. Alien DNA. Yes, 3 names are there. If you find these 3, any one word, you remember we are talking about nothing but gene of interest. Generally it is called gene of interest, or the another name for gene of interest is foreign DNA. Another name for foreign DNA is alien DNA. Any word if they given, you just remember alien DNA means nothing but gene of interest, foreign DNA means nothing but gene of interest. Yes, gene of interest we got. Now what we have to do with it? Now we have to make the so many number of copies of this gene of interest. Now how many number of copies you having? Only one gene of interest you having, so sad yaar. You have only one gene of interest. Yes, what can I do with this one gene of interest, sir? I want so many number of gene of interest. How can I get this so many number of gene of interest? Means there are the 2 techniques. To make this gene of interest many, to make multiple gene of interest, there are 2 techniques are there. Number 1, you can multiply this gene of interest. You can multiply this gene of interest by a artificial technique. Artificial technique, it is not a natural, it is artificial. Artificial means what? Which happening inside of a machine, which happens inside of a machine. Yes, which happens inside of a machine. And another one is natural process or a natural technique. Natural technique means which is happening inside of a cell, which happens inside of a cell. Later we will discuss it. Means at present you have only one gene of interest, but you want many copies of gene of interest. Yes, you can make many copies of gene of interest by 2 ways. Number 1 is artificially inside of the machine, and you can make naturally inside of the cell. Okay, you can make naturally inside of cell. In these 2, which one is most preferable? The most preferable thing is natural technique is very far better when compared with the artificial technique. Why? Because natural is natural. Na? How many machines we are developing? Natural food is natural food. Whatever we are eating outside, that is not at all natural, like junk, fast food, all that. They are not natural. Na? Natural is natural. When compared with these 2, in these 2, which one is the best one? Means natural. Natural technique is very. So gene of interest we have to multiply by 2 ways, artificially, naturally. Now come to the artificially. Artificially means which is happening inside of machine. Yes, that technique is called PCR technique. That technique is called PCR technique. Polymerase. PCR technique. That is PCR technique. What is the meaning of PCR? Polymerase chain reaction. What is this PCR means? Polymerase chain reaction. There are so many reactions are happening. Each and every reaction is linked with a chain, means one reaction is followed by another reaction, another one is followed by another one, and this is happening, this all chain reaction is happening in the presence of a one enzyme. In the presence of a one enzyme. What is that enzyme name? Polymerase enzyme. It is all happening inside of a one enzyme because of that only polymerase enzyme. This polymerase enzyme is making the reactions. Those reactions are in the form of a chain. Because of that only this machine technique is called polymerase chain reaction. Simply in shortcut we call it as PCR. For what purpose? For copying purpose. For copying purpose. Copying the gene of interest. At present you have how many gene of interest? Only one. So we have to multiply. Yes, that multiplication is also called amplification. Amplification means nothing but copying. Amplification. Amplification. In your bio textbook they have given amplification of gene of interest by PCR. Amplification means nothing but copying. Amplification means nothing but copying. Yes, how it is going to happen? I told that it is happening in a chain of reaction, and this reaction is happening in the presence of an enzyme. The enzyme name is polymerase. So it is polymerase chain reaction. See, remember this PCR technique. To complete this PCR technique, there should be a high temperature. High temperature is required. This is happening in high temperature, not in low temperature, which is of approximately above 50 degrees Celsius. 50 degrees Celsius in high temperature the PCR technique is happening. Okay sir, fine. In PCR technique, first of all, how many steps are there? How many steps are there? In PCR technique, polymerase chain reaction technique, in polymerase chain reaction technique, there is a presence of 3 steps. 3 steps are there. Within the 3 steps, one PCR will be completed. One copy will be made. One gene of interest, if undergone one PCR technique, another copy is formed. Another one, 2 copies. Like that. First one is denaturation. Important. Denaturation. Yes. Denaturation is the first step, first process in the PCR technique, and this denaturation is happening in the presence of 95 degrees Celsius temperature, which is happening in high temperature. What is denaturation? I will tell that. What is denaturation? I will tell that. Next one, after denaturation, the second event is annealing. A-N-N-E-L-I-N-G, annealing. And this annealing is happening in the presence of approximately 55 degrees Celsius, which is happening inside of 55 degrees Celsius. So we have to raise the temperature. When you raise the temperature to 95 degrees Celsius, denaturation will happen. And later you have to decrease the temperature up to which level? 55 degrees. At that time, what is going to happen? Annealing is going to happen. After annealing, what is the third event? The third event is extinction. Extinction. This is the third event, and the extinction is happening in the presence of approximately 72 degrees Celsius, which is happening approximately in 72 degrees Celsius. In the 2020, they asked this question: arrange the events in the PCR technique. First one is the denaturation followed by annealing followed by the extinction, and which is happening at what temperature? You remember this. You remember. These are the 3 processes. Okay, let me explain what is denaturation and what is annealing and what is extinction. Let me explain that one. Before explaining this, first of all, sir, to perform the PCR, what is required, sir? To perform this PCR, to do this polymerase chain reaction, what we require, what are the requirements to do any work? We require some requirements, right? For example, I am taking the online class. Means I require a marker, I require a camera, and I require a one board, and I require a one doom light. These are the requirements. Same like that only, we have to copy this gene of interest. For this copying this gene of interest, we choose the artificial technique which is known as PCR. So what is required in this means, let me erase this. Requirements is Number one, first thing that is required is enzyme is required. One enzyme is required. What is that enzyme? That enzyme name is DNA polymerase. DNA polymerase. S means what? Enzyme. It is making the polymer. Which polymer it is making? DNA polymer it is making. So the enzyme name is DNA polymerase. As of I told, enzymes are given the name based upon their function. First thing is DNA polymerase. Sir, in this high temperature, enzymes will not perform function. No, enzymes will get damaged. Enzymes are inactivated. In biomolecules we read enzymes are working only under certain condition. Now this is the DNA polymerase enzyme. In this high temperature, can it tolerate? No, it can't tolerate. Because of that only, we should not take the human's DNA polymerase enzyme. We have to take the DNA polymerase enzyme which is present in the bacteria. Yes, for example, this is the bacterial cell. Inside of this bacterial cell, there is a presence of enzyme. What is that enzyme means? DNA polymerase enzyme. See, very dangerous DNA polymerase enzyme. So it is a DNA polymerase enzyme. What is this bacteria name, sir? What is this bacteria? This bacteria is present in higher temperature. Higher temperature. Thermophilic. Thermo means what? Temperature. Thermometer, a instrument responsible to measure the temperature. Thermo means what? Temperature. So this bacteria is a thermophilic bacteria which can present in higher temperature. So what is this bacteria name? The bacteria name is Thermus aquaticus. The bacteria name is Thermus aquaticus. Let me write here. The bacteria name is Thermus aquaticus. Thermus aquaticus. You should remember, Thermus is the genus name, Aquaticus is the species name. So you know, genus name starts with capital letter, species name starts with a small letter. So now you remember, this bacteria contains enzyme. What is the enzyme? DNA polymerase enzyme. And this enzyme is resistant to the higher temperature. Resistance to higher temperature. So this DNA polymerase enzyme is called with another name. What is that another name means? Talc polymerase. Talc polymerase enzyme. What is the meaning of this talc polymerase? Baba, simple ra. In thermos, what is the first letter? T means here. T stands for thermos. In genus, what is the first two letters? AQ. AQ means what? Aquaticus. And what enzyme is this? It is making the polymer or monomer? It is making the polymer. So it is a polymerase. So simply we call it as talc polymerase. Talc, that indicates T stands for thermos, AQ stands for aquaticus. So the entire name is what? Thermus aquaticus enzyme. And this enzyme is present in the thermus aquatic bacteria which can survive in the higher temperature. So now this enzyme is resistant to the higher temperature. Which resistance to the higher temperature? Yes, fine. After, along with this bacteria, let me erase this. Along with this enzyme, okay, we got this enzyme from Thermus aquaticus. And along with that enzyme, we require two sets of primer. We require two sets of primer. Two sets of primer. Primer. What is this primer? Now, primer is a small molecule. Which molecule means RNA molecule? Remember that. Oligonucleotide. What is the meaning of oligonucleotide? Oligo means 5. Oligo means 5. It is having the 5 nucleotides. 5 nucleotides. So that is not DNA, that is RNA. So just let me represent the two sets of primer. For example, this is the RNA primer number 1. RNA primer number 1, which having the nitrogen base. You can write anything, adenine, guanine, cytosine, anything you can write. This is the set number 1, and we require one more set na? Yeah, this is the one more set. Another RNA primer. Another RNA primer. You remember, primer means nothing but oligonucleotide. Which oligonucleotide? DNA na RNA na? RNA. Yes, how many sets are required? 2 sets of primer is required. And along with these primers, there is a requirement of nucleotides. Which nucleotides? Free nucleotides are required. Free nucleotides tides are required. You know, nucleotide is made up of how many components? 3 components. I hope you know. Number 1 is what? Pentose sugar. And the second component is what? Nitrogen base. And the third component is what? Phosphate. These 3 components we call them as nucleotide. It is what bond? N-glycosidic bond. And here what bond? Phosphoester bond. This you will get in the molecular basis of inheritance. While I am teaching that chapter, and I will clearly explain what is this pentose sugar, what is this nitrogen base, and what is this phosphate. Okay, fine. Free nucleotides are required. How many number of nucleotides? So many number of nucleotides are present. Yes, required. These are the requirements. These are the requirements to successfully complete the polymerase chain reaction. Take one screenshot if you want, please. Yes. What are the requirements? DNA polymerase enzyme. You are getting this enzyme from where? Bacteria. So the enzyme name is what? TOC polymerase. T means thermos, AQ means aquaticus, which can function in a higher temperature. Next we require what? 2 sets of primer. Primer is a what molecule? RNA molecule. Oligo means 5. It will have 5 nucleotides. Next one, along with that we require what? Free nucleotides. So the PCR technique is followed within how many steps? 3 steps. Denaturation, annealing and extension. Yeah, let me tell this now detailedly. What can I do here? Where have to? So here I can erase. Yes, we have taken the PCR machine. Why? Because why all we doing? To make the copy of gene of interest. To make copies of gene of interest. What is the gene of interest? Nothing but DNA molecule only, na? We got it from where? DNA. Isolation of DNA, cutting of DNA, taking the fragment of DNA. Yes, this is what molecule? Nothing but DNA molecule. For convenience purpose, let me write that DNA molecule somewhat large. This I am writing here. This gene of interest, which we call it as a foreign DNA, which we also call alien DNA, I am representing here. Yes. What is this? Yeah. This is the strand number 1 and this is the strand number 2. You know that if one strand having the 3 prime, the another strand will have 5 prime. And here 5 prime means this strand will have 3 prime and this will be 5 prime. Anti-parallel. The 2 DNA strands are what? Anti-parallel. Now we remember it contains what here? Nitrogen bases. Just let me represent only few nitrogen bases. Here adenine, next here guanine. I think, can you see this? Yes, clear. Yes, adenine, guanine, and next I can write cytosine, and I can write thymine, and next adenine, next cytosine, next guanine. So complementarity. Adenine always make complementarity with whom? Thymine. And it make with whom? Cytosine. And it will make with whom? Guanine. Thymine will make adenine. Next here thymine. Next guanine. Next cytosine. And you know, in between the guanine and cytosine, how many hydrogen bonds? 3. How many hydrogen bonds? 3. In between adenine and thymine, how many? 2. In between adenine thymine, how many? 2. Guanine cytosine, how many? 3. Guanine cytosine, how many? 3. Adenine thymine, how many? 2. Yes, these are the hydrogen bonds. Now this DNA molecule, we throw it inside of a machine, PCR machine. And along with this, let me represent a imaginary diagram to understand. It's an example, okay? It's just an example. Let me represent. This is, you imagine that this is the PCR machine. In this PCR machine, I throwed a DNA molecule. Along with the DNA molecule, I added the enzyme. What is the enzyme? TOC polymerase enzyme I added. And along with that, I also added the 2 sets of primer. And after 2 sets of primer, and I also added the so many number of nucleotides. All these small dots, let me represent with another color. All these small dots is representing the free nucleotides are present. Can you able to see diagram clearly up to bottom? Yes. All I added in the machine. What machine is this? PCR machine. And what I did, I started heating this. I started heating this temperature. I started heating. Up to what level I have to eat? I have to heat. You know what is the first step in the PCR polymerase chain reaction? The first step is denaturation. Denaturation. And for denaturation purpose, how much temperature is required? 95 degrees Celsius temperature is required. Because of that only I am heating, increased the temperature and finally reached to 95 degrees Celsius. Whenever 95 degrees, it is reached to 95 degrees Celsius, what is happening? The hydrogen bonds which are present between the nitrogen bases, the hydrogen bonds which are present between the nitrogen bases, these hydrogen bonds are broken down. Whenever these hydrogen bonds are broken down, what is going to happen? This stand is coming. These two strands got what happened now? The two strands got separated like this. The two strands are separated. This is called denaturation. 5' and 3'. And I represented the nitrogen bases. Let me represent those nitrogen bases. Here what? Thymine. So here it will be adenine. Next guanine. Next cytosine. Next thymine. Here thymine. So it is adenine. Guanine. It is cytosine. It is cytosine. It is what? Guanine. Yes. What is denaturation? What is denaturation? Denaturation means nothing but separating, breaking down of hydrogen bonds and separating the two DNA strands. This is called denaturation, and this denaturation is happening in how many degrees Celsius? 95 degrees Celsius. Okay, two DNA strands are what happened? Separated. Now this DNA strand have the ability to synthesize new DNA strand here. So it is acting as what? A template. It is acting as template. And next, this DNA strand also have the capability to develop the new DNA strand. So this strand is also acting as what? Template. So both the separated DNA strands are acting as templates. Acting as what? Templates. Now, second step. After denaturation. What is the second step? The second step is annealing. ANNELING. Annealing. To complete this annealing, the temperature should be how much? 55 degrees Celsius. 55 degrees Celsius. So what I did, I am decreasing the temperature. No, this much temperature is there na. Two DNA strands are separated na. So let me decrease this temperature. And decrease and reach it to how much? 55 degrees Celsius. And whenever I reach it to 55 degrees Celsius, what happening? There is two sets of primer na. Let me represent the primers with black. Yeah, this is the primer number 1 and this is the primer number 2. Two primers are there. Where these primers will go? Primers is moving. Where these primers are moving? What this wire here? Wire is disturbing me completely. Where these primers are moving, whenever I decrease the temperature, means when I reach to 55 degrees Celsius temperature, the two primers which are present in the machine, so here the DNA strands are what happened? Separated. I representing there na. The two DNA strands are what happened? Separated like this. Like this, the two DNA strands are separated. And I represented there. This is what prime? 3'. It is what prime? 5'. And it is what prime? 3'. And it is what prime? 5'. Now, where these two sets of primers are moving, the primer will always reach towards 3'. Okay, fine. Where 3' is present? For this stand, 3' is here. So the primer is going to fix in this place like this. Like this, the primer is attached. Here 3' means here will be what prime? 5'. Here will be what prime? 5'. This is the primer number 1. This primer number 1 have complementarity. Yes, here what is there? Thymine is there. So here it will be adenine. Here what is there? Cytosine is there. So here it will be guanine. Like this. This is the primer number 1. And one more primer is there na? One more primer. That one more primer is moving to another strands 3'. Yes, that one more primer is reached here. This is the one more primer which is reached here to the 3'. 3' for DNA and it is the 5'. 5 prime. Why? Because DNA have to be in anti-parallel, na? So the two sets of primers and complementarity, it will make complementarity. Here cytosine, na, here it will be guanine. Here guanine, na, here it will be what? Cytosine. Complementarity is more. This process of setting of primers at the third pre-prime, the process of moving of primers, they are present in machine, right? Now these, when I decrease the temperature, the two primers are moving towards which prime? 3 prime of the DNA. For this stand it is moving to 3 prime, and for this stand it is coming to 3 prime. This moving of primers to the 3 prime, this process is called annealing process. That process is called annealing process. Yes, that is annealing process. After annealing, what? Nothing but extension. Nothing but what? Extension. The third event is what? Extension. E-X-T-E-N. Extension. This is the extension. To do this extension, once again I have to raise the temperature. Yes, at how many degrees Celsius it is happening? As I told, approximately 75 degrees Celsius. 75 degrees. Actually here how much degrees now? 55. So once again I started raising the temperature and reached it to how many degrees Celsius? 75 degrees Celsius it is reached. Whenever 75 degrees Celsius it is reached, now our big boss is coming. Who is that big boss? Polymerase is present, na? Yes, see here, we also added the enzyme, na? What enzyme is this? Talc polymerase enzyme. This is our big boss. My function is, now I have to do the function what? Extension. Yeah, the big boss is coming. Here it is there. And this big boss contain a big bag. A big bag it contains. Big bag. Yes, in this big bag what are present? All these are what I told. Free nucleotides. Yes, here there is a presence of free nucleotides are present. Yes, free nucleotides. It may be adenosine, or it may be guanosine, or it may be thymidine, or it may be cysteine. In this manner, 4 free nucleotides are present inside of this bag. Inside of that bag. What is this doing now? Checking here. Yes, what is present here? Guanine is present. So what I have to fix here? So the enzyme it have to fix here is, by extending this, it have to fix the which nucleotide? Cytosine. After that it is moving. Enzyme is moving in this manner. From 5 prime to 3 prime it will move. It is what prime? 5 prime. It is what prime? 3 prime. So it is moving. Okay, here adenine, so nucleotide thymine come and sit here. And here thymine, oh nucleotide, here come adenine, sit here. And next here guanine, yeah, come cytosine, cysteine come here. And next here cytosine, oh guanosine come and sit here. And this manner it is ended, and this is what prime? 3 prime. So what happened? It extended the primer. And it extended the primer by adding the nucleotides, by checking the complementarity of DNA strand. And after that the same enzyme comes here. And once again it started what? Extension. It started what? Extension. Yes. Okay. Enzyme is seeing what is here? Adenine. Okay, then come. Thymidine, come and sit here. What is there? Next, thymine. Okay, come and sit here. Adenine, adenosine. Cytosine. Oh, guanine, come and sit here. And next, guanine, cysteine, come and sit here. And here is what? Adenine, thymine, come and sit here. In this manner, what it done? Extension. From which prime to which prime? 5 prime to 3 prime. So the enzyme always code only in one direction. The polymerase enzyme always synthesize the new DNA strand or new RNA strand only in one direction. From which prime to which prime? From 5 prime to 3 prime. 5 prime to 3 prime. Yes. Initially, we have one piece of gene of interest. Is it? Now, after completion of one cycle, see, we have 2 gene of interest. We have 2 gene of interest now we have. One became how many? 2 gene of interest it is happened. And further you added the one more sets of primer and so many number of free nucleotides you are adding, and once again you raised the temperature. You raised the temperature up to what? 95 degrees Celsius. When you raised it to 95 degrees Celsius, these 2 DNA strands what happened? Separated. These 2 DNA strands what happened? Separated. At that time what it will be? See, this is okay, black na, let me represent here. Denaturation, black. What prime? 3 prime. What prime? 5 prime. Next, what color I added? I added the, okay, red color I added na. Okay, fine. This is the red color. this one. So this is also in black only, na? Okay, fine. Let me 3 prime, sorry, separated, na? It is separated. Whenever it is separated, 5 prime and 3 prime, these DNA strands are separated. Now same like that only these two DNA strands are also what happened? Separated. Yes, I represented it is with what? Green. Which prime? 3 prime. Which prime? 5 prime. And next is what? Red I represented. So I think you can't see this one. Separated. Two DNA strands are what happened? Separated. Each and every DNA strand is acting as what now? Template. Denaturation is completed. Two DNA strands are separated. Now once again I am decreasing the temperature to 50 by annealing. Annealing means what? Setting of primer. Primer always moves to which prime? 3 prime. Here one primer, here one primer. Same, here one primer, here one primer. After annealing, once again I raised the temperature. What? Extension. By using the enzyme TOC polymerase and with the free nucleotides, once again there is a synthesis of new DNA strand like this. So DNA strand number 1, DNA number 2, DNA number 3 and DNA number 4. See, initially we have 1. After one PCR we got 2. After second PCR we got 4. In this manner 4 will become 8. 8 will become 16. 16 will become 32. 32 will become 64. In this manner if you continue the PCR technique approximately for 30 cycles. If you continue this for 30 cycles, approximately you will get 1 billion copies. 1 billion copies you will get if you continue for 30 cycles. So just I continued only how many cycles here? 2 cycles. First cycle we got 1 DNA2, 2 we got. Next another cycle 2 DNA2 we got 4. Only just 2 cycles I did. For 2 cycles I got 4. In this manner third cycle 8. Fourth cycle 16, fifth cycle 32. In this manner if I continue for 30 cycles, approximately I am getting how many copies of gene of interest? 1 billion copies of gene of interest that I am getting. Yes, see, initially we have how many gene of interest? Only 1. When we add this when we done this PCR technique, how many gene of interest we got? 1 billion, not in million. For approximately, for how many cycles? 30 cycles. 30 cycles. Yes, just this is very important question in the NEET and as well as in CET and as well as for the PUC examination. It is a very important. Very in NEET most of the time they asked the arrange the steps or events in the PCR technique. Denaturation followed by annealing followed by the extension. The enzyme TOC polymerase enzyme is functional in which event? Extension event only. In extension event only enzyme is coming and extending that primer. Next one, temperatures you remember 95, 55 and 75 degrees Celsius. The enzyme TOC polymerase and it is extracted from the bacteria. What the bacteria name? Thermus aquaticus bacteria, which is a thermophilic bacteria. Yes, this is the method number 1. This is method number 1 that we can make the so many number of copies of gene of interest or foreign DNA or alien DNA. And next one is what? Natural. Next one is what? Natural. Yes. Now let's discuss about this natural technique. Let me erase this here. I hope you understood this. If you want, you can take the screenshot. Please subscribe to the channel, and if you understanding properly, so please comment and like and share the video. Please remember the name, channel name. I no need to say, you know that. Please do subscribe and like and share. Any doubt, comment me and comment me how the class is even. Next, natural method. How we can increase the alien DNA or foreign DNA. Let's see now. Artificially we prepared. Naturally. This natural is somewhat complicated. It is one by event, one event after one event. It takes some confusion. It will give some confusions. Okay. But don't worry, I will clear your confusions. So just listen carefully. Just listen carefully. Now, copying the gene of interest. I don't like that artificial. I don't want gene of interest, copies of gene of interest which happened artificially. I want gene of interest which it will get naturally. I want. Okay na? Okay then. Let's see. Let me write our gene of interest. Yeah. This is our gene of interest. Nothing but foreign DNA or alien DNA. Everything we are discussing is to prepare which DNA? Recombinant DNA. Don't forget this. All this we are doing to prepare this which DNA? Recombinant DNA. Recombinant DNA. Okay. This is the gene of interest, foreign DNA or alien DNA. Naturally, na? Natural technique means which is happening inside of cell. Which is happening inside of where? Cell. Which cell? Bacterial cell. Which cell? Bacterial cell. Prokaryotes. Bacteria are what? Prokaryotes. Why? Because they are not having the nuclear membrane, right? So in bacterial cell it is happening. Why, sir? In bacterial cell? Why in bacterial cell? Yeah. We want copies, na? What we want? Copies we want. We want copies of it. We want copies of it. Yes, we want copies. For that purpose, we have to select the bacteria. Just imagine this is our bacteria. Bacteria. And you know, the plasma membrane of the bacteria having the infoldings. Infoldings. Like this, there is a presence of infoldings. And these infoldings having the different shape, filamentous, and it will perform total 7 functions. In cell, the basic unit of life it is there. You tell me these infoldings are what? Comment me please. Okay, yes, tell me name of these infoldings. You tell me. Bacterial cell having the cell membrane, that cell membrane having the infoldings. Foldings are inside, na? So what is the name of this infoldings? And that infoldings perform 7 functions. So please comment that. Yeah, this bacteria contains DNA. Yes, this is the bacterial DNA. The bacterial DNA is what? Naked DNA, which is not covered with the nuclear membrane. So it can able to see with eye, with our naked eye we can see. So it is a naked DNA. Okay, fine. And along with this, bacterial cells contains, most of the bacterial cell contains additional DNA like this. Additional DNA like this it is present. These are the additional chromosomal DNA. It is a chromosomal DNA. It is present inside of chromosome. We are not talking about this DNA. Along with this chromosomal DNA, there is a additional DNA is present. Apart from chromosome, additionally it is present. So additional chromosomal DNA like this. And this DNA is double stranded. See, 2 circles I have given. First of all, it is circular or not circular? Yes, it is circular. It is what? Circular. Circular. It is a double stranded or single stranded? Yes, of course, it is a double stranded. Double stranded. Circular double stranded DNA. What is the name, sir? What is the name? The name we call them as plastids. These we call them as plastids. What are plastids? Plastids are circular or double circular DNA molecules, point number 1, and they are the extra chromosomal DNA. This is a chromosomal DNA. Along with the chromosomal DNA, extra they are present. Extra they are present. So because of that only, these are extra chromosomal DNA. Extra chromosomal DNA. Sir, what will happen if I remove these plasmids? If I remove all these plasmids, can this bacteria cell can survive? Of course, yes. Why? Because it is having its own chromosomal DNA. Na? If it is present, no problem. If it is absent, no problem. So these plasmids are not essential for bacteria. What is essential? If these are removed, if bacteria is dying means, oh, no, Baba, I won't give, I want those plasmids. Why? Because I will die. At that time, what essential they are. But if I remove these plasmids from the bacterial cell, it won't get, nothing is happening. So these plasmids are non-essential. The bacterial cell doesn't require those. So these are non-essential DNA molecule. Non-essential DNA molecule. Yes, and these plasmids are important. Resistance to the antibiotics. Resistance to antibiotics. Resistance to antibiotics. See now, and these plasmids, most of the plasmids are used as vectors. As I told, vector means which helping in the transferring of something. For example, I want to move from here to somewhere, means I will go in the vehicle, in car or in bike or in bus. So the bus, it is transporting me. So the bus is the vector for me. Vector means what? Something it will transport from one organism A to organism B. This I taught in my past lecture. Yes, in the previous lecture. Plastids. 1 minute. It is not plastids here. It is a plasmid. It is a plasmid. Sorry. How much wrong it is? Plastid is chloroplast, leucoplast, and one more chromoplast. In cell, the basic unit of life, in plant cell, we will get one cell organelle, plastids. That plastids 3 types, na? Chloroplast, chromoplast, leucoplast. That is a plastid. It is not plastid here, it is a plasmid. How much mistake I did? Plasmid it is. Plasmid. Plasmids are acting as vectors. Plasmids are double circular DNA molecule. Plasmids are extra chromosomal DNA molecule, and plasmids are non-essential molecules, and plasmids are resistant to antibiotics. Resistance to antibiotics. Yeah, fine. Yeah, human, yeah. General mistakes are common, na? Human. Yes, I am not a robot, I am not an alien to say or everything is correct, na? So sometimes mistake is just correct. Now let's see. Plasmids. Circular DNA molecule. Okay, fine. These plasmids contain, one more important point I forgot to. These plasmids can self-replicate. They can undergo replication. Yes, this is the very important point. DNA means what? Which have the capability to undergo replication. So now this is nothing but what DNA? Circular DNA. So this DNA also have the capability to undergo replicate without permission of anyone. So this plasmid no need to ask chromosomal DNA. Okay, I want to replicate. Give permission. I will replicate. No, not required all these. Who are you, man? I can replicate myself. It means the plasmids are showing the self-replication. They can undergo self-replication. This is important point. You remember all these. Plasmids can act as vector. Plasmids undergo self-replication. They are double circular DNA molecule, extra chromosome, non-essential, and resistant to antibiotics. Okay, fine. I told that these are acting as vectors, right? Okay, vector. This vector, after what we have to do means, for example, where is our gene of interest? Yeah, this is our gene of interest, right? This is our gene of interest. You keep that gene of interest here, This vector having the one site, there is a one site is present. One site, that site is called ORI site, origin of replication site, where replication is originated. Why? Because plasmids undergo self-replication, na? So there is a one site, that is the one site where what is started there? Replication. So origin is started. So ORI site, origin of replication. Yes, what will happen? It will replicate, and this plasmid will give one more plasmid. And another replication, it will give one more plasmid. And this plasmid undergo replication and it will give another plasmid. And it will undergo replication and it will give. In this manner, what happening? plasmid number is doubling. After replication, one will give 2, and this will give in the one more 2, and this will give 2. In this manner, what happening? Plasmid number is increasing. Means what? Yeah, that is a doubling, na? Doubling means what? Which are exactly looking like means what? Clone. What is we called as clone? What is clone? My clone it is. Means my body double. There is a my body double who exactly looks like me. It means I am original and this is my body double. Means this is what? Cloning. What I did cloning here. So only my body double. So exactly we are getting these so many plasmids. Plasmids are acting as what? Vectors. And all these plasmids have the ability to undergo self-replication, and they can give these so many plasmids and they can clone. Because of that only these vectors we call them as cloning vector. Cloning vectors. Yes, this is the heading. That is the heading. Cloning vectors. Vector means nothing but what? A plasmid. Vectors are of two types of vectors are plasmids and bacteriophages, which can get a double, which can get a clone. So we are discussing about cloning vectors. Plasmid and bacteriophages. Two are the vectors. Okay. Yes, this is the cloning vector. Now let's see. They can get a clone. That's all here. Apart from that, these cloning vectors, number one is plasmid. Number one is plasmid. And number two is bacteriophage. Bacteriophage. You know very well, bacteriophage is the virus. That virus inside of the bacterial cell, it can double. It can replicate. This is the most commonly used cloning vectors are plasmids and bacteriophages. Plasmids are present in bacteria and bacteriophages is nothing but a virus. Now we remember some plasmids, this is the plasmid, okay. I throw this plasmid inside of one bacteria. At the time this plasmid is replicating only one time, means it is having only one ORI site. So if it is having only one ORI site, it will give only one copy. If you want many copies, that plasmid should contain many ORI sites. If it is having the many ORI site, it will give the so many multiple number of plasmids it will give. Means if you want more copies of gene of interest, you have to add that gene of interest with the plasmid which having more ORI sites. At that time, how many ORI sites it having? Approximately maximum plasmid contain 100 to 150 ORI sites. If you tag the gene of interest to that plasmid, it will give 100 gene of interest or 150 gene of interest. When you compare with the plasmid, bacteriophage will give this so many number of more and number of copies it will give. You don't get confused. One thing you remember, cloning vector means a vector which can get a clone, double. 2, plasmid and bacteriophage. 2. If you want more gene of interest, you should find the plasmid which having more ORI site. If you, for example, this is the plasmid, it have the 100 ORI site. If I tied the gene of interest here, at the time what will happen? It will replicate, self-replicate, independently it will replicate. Na? It is having how many ORI sites? 100. So you will get how many copies? 100 copies you will get. 100 copies. If you want only one copy, at that time you can tag that gene of interest only to the plasmid which having only one ORI site. It will give only one copy. Okay. Now let's see. Regarding plasmid is important. Take a screenshot if you want. Regarding plasmid is important. Now you remember, nowadays plasmids are also used in the engineering technique to identify the non-recombinants and recombinants. Non-recombinants and recombinants. This is the most confusing part where most of the students will face difficulty. Four names. Transformants and non-transformants. Be careful. 4 names I said. Let me erase everything here. I step on to the concept now, which is confusing more. Yeah, we are preparing the recombinant DNA. Na? So let me, nothing, recombinant DNA is nothing, okay? Just listen. Just listen. I will prepare the recombinant DNA. This is the vector, a double circular vector, double standard DNA molecule. And I cutted this vector with the enzyme. I won't cut it this scissor, na? With the enzyme what? Restriction endonuclease enzyme. As it cutted here. This part is acting as glue. Glue means what? Gum. Okay, this part is acting as what? Glue, acting as gum. And this part is also acting as what? Glue, gum. And next we have our gene of interest. We have our gene of interest. Yes, this is our gene of interest. This is our gene of interest, which contain ends. And here it is having the glue. It also contains what? Glue. Now immediately what will happen means this gene of interest, whenever we cutted this, the gene of interest is joined here. Joined here. Like this is joined. Now you remember, this is our foreign DNA or gene of interest or alien DNA. And this red color is what? Plasmid's DNA or vector DNA. This is now called it as recombinant DNA. It is called recombinant DNA. We cutted the DNA and we combined the DNA. Because of that only it is a recombinant DNA. So recombinant DNA contains plasmid's DNA. Along with the plasmid's DNA, it also contains what? Foreign DNA or alien DNA or gene of interest. How it is going to happen? Just remember, in the test tube, there is a presence of so many number of plasmids. I added all these are the plasmids. All these are the plasmids. For example, I can't draw all the plasmids. So many plasmids are present and all of them having the sticky ends. All these are the sticky ends which having the gum. Which acting as glue. And I added the gene of interest. I will add only one gene of interest. Okay, this is our gene of interest. Now this gene of interest got joined here, and this is called what DNA? This is called recombinant DNA, which is rDNA. Yes, this is ended, but there is no furthermore gene of interest now. And immediately these two ends are also acting as what? Glue. Immediately these two are what happened? Joined. Okay, fine. See, they made mistake to us. They're fooling us. See, they got separated, they got cutted with restriction endonuclease, and they didn't attach with the gene of interest. They immediately get joined. So these are recombinant DNA here. I mean, here gene of interest present or absent? Absent. This is called non-recombinant. It is called non-recombinant. Non-recombinant means nothing but which doesn't contain gene of interest, which contains only plasmid DNA. Then recombinant DNA means which contains gene of interest. Which contains what? Gene of interest. Yes, this is about recombinant DNA and this is non-recombinant DNA. This is the confusion clear. In recombinant DNA, gene of interest is present, and in non-recombinant DNA, gene of interest is absent. Now, next step. What is the next step? Next step is a bacteria cell number 1 and a bacteria cell number 2 and a bacteria cell number 3. Like this, 3 bacterial cells are present. So now we remember what is the transformants and what is non-transformants now. What is the transformants and what is non-transformants? Now what I did means, now all this is happening in bacteria. We can't see all those. Scientists observed and they are telling, that's all I am discussing. We are discussing separately, but all this happens simultaneously. Now what happened means, this is which DNA? Recombinant DNA. This recombinant DNA is entered, taken up by this cell, bacterial cell number A, and you consider this is the bacterial cell number B, bacterial cell number C. This DNA is entered. Yes. Which DNA is this? Recombinant DNA. It is which DNA? Recombinant DNA. If it is taken this recombinant DNA, it is transformed. It taken something, na? It taken something. So this is transformed. Next, this B bacterial cell also taken this circular DNA, but it is taken the which DNA it is taken? Non-recombinant DNA. Means here gene of interest is absent, but it is taken, so it is also transformant. It is also what happened? Transformed. It is also what happened? Transformed. Transformed. Both are transformed. Why? Because both of them contains circular DNA. Both this bacterial cell A contains circular DNA and it is also containing circular DNA. But here gene of interest is present. Which DNA it is having? rDNA it is having. So it is recombinant DNA transformed bacterial cell. Full name. Recombinant DNA transformed bacterial cell. It is the bacterial cell. It is transformed. Transformed means what? Changed. How it is changed? It is changed by taking the rDNA. So it is recombinantly transformed bacterial cell. Now it is also what happened? This bacterial cell also is transformed. This bacterial cell also what happened? Transformed. Yes, but it is transformed with a non-recombinant DNA molecule. So this bacterial cell is non-recombinant transformed bacterial cell. You see? It is not taken anything, na? So it is not transformed. Non-transformed bacterial cell. Hey, don't get confused. Don't get confused. These two bacterial cells taken the DNA. So they are transformed. They are what? Transformed. It taken which DNA? Recombinant DNA it is taken. So it is recombinant DNA transformed bacterial cell. It is taken the DNA. Which DNA? Non-recombinant. So non-recombinantly transformed bacterial cell. It doesn't take anything. It doesn't take anything means it won't get transformed, na? So it is what? Non-transformed bacterial cell. Now in these three cells, cell A, cell B, cell C, which cell we require? Our work is because of for what? Gene of interest. We don't want non-transformed bacterial cell. Why? Because we don't want this. Why? Because here gene of interest is absent. It is not even taking the circular DNA. And it is transformed. It is transformed by taking the DNA. But in this bacterial cell, our gene of interest is not there. So we don't want this also. We don't want non-transformed also. So we want the recombinantly transformed bacterial cell. This is we want. Selection. Sir, it is board. You can write this. Okay, these two are transformed by taking the circular DNA. It is not transformed. It is not taking the circular DNA. Na? You can write on the board. But how can we select? How can we select? Because of that only next heading is selectable markers. Selectable markers. These selectable markers are also present in vector. Who is the vector that we have taken? Plasmid. Plasmid. So, for today's it is enough. You understand carefully that a non-recombinant DNA. What is a non-recombinant DNA? What is recombinant DNA? What is a transformed bacterial cell and what is a non-transformed bacterial cell? Non-transformed bacterial cell means which doesn't take the vector. We don't want that. Transformed DNA molecules means two are there. Sorry. Transformed bacterial cells means two are there. One is recombinantly transformed bacterial cell and another one is non-recombinantly transformed bacterial cell. Non-recombinant means what? Their gene of interest is absent. We don't want that. Recombinant bacterial cell means what? Bacterial cell which contain recombinant DNA means which contains. This is we want. How can you select? How can you select? For that purpose only we have selectable markers. In the next class, I will discuss about these selectable markers and I hope you got some idea about this now. You got some idea regarding PCR and regarding this cloning vectors. So please, after watching this class, so comment, like, and if you're not subscribed to my channel yet, so please subscribe, and immediately after uploading my video, you will get the notification as much as fast. So please subscribe to the channel and like, and don't forget to comment that any doubts you can ask. How was the class? Give the feedback. And thank you for all. Thank you.